Now that we have done with the entire chapter, we go to the last part, certain experiments which were conducted to prove that the DNA replication is by the semi conservative method of replication, and certain experiments to prove that the DNA is the genetic material. So, which were these experiments of genetics? Let's finish the chapter by doing this part. While proposing the double helix structure of the DNA, Watson and Crick had immediately proposed a scheme for the replication of DNA. They had stated that it has not escaped our notice that specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That was proposed by Watson and Crick in 1953. And what is the meaning of this? The scheme suggested that two strands would separate and act as a template for the synthesis of new complementary strands. The two strands would separate, each strand will act as a template and help in the synthesis of new strands. After completion of replication, each DNA molecule would have one parental and one newly synthesized strand, which was called as the semi-conservative method of replication. One parental strand will be having one new strand. One parental strand will be having one new strand. So in each daughter DNA, there is one parental strand. In each daughter DNA, there is one parental strand and one is a new strand. One is a new strand. This is called as the semi-conservative DNA replication. The experimental proof that DNA replicates semi-conservatively was first shown in E. coli by Messelson and Stahl, who performed an experiment in 1958. Now, what did they do? They grew E. coli in a medium containing N15H4Cl. N15H4Cl, N15 is a heavy isotope of nitrogen. Elementary nitrogen is N14. N15 is a heavier isotope of nitrogen. तो N14 normal होता है. They grew E. coli in N15. And for many generations, they multiplied N15. The result was N15 was incorporated in the newly synthesized DNA as well as nitrogen containing compound. So, all the strands of the DNA will have N15. जितने भी बैक्टीरिया है, बैक्टीरिया के अंदर N15, N15. So, suppose this is a bacteria and बैक्टीरिया का ये strand है. Both strands of the bacteria have radioisotopes N15 in them. So both the strands are N15, N15. And this heavy DNA molecule could be distinguished from a normal DNA by centrifugation in a cesium chloride density gradient. तो आपको जब डीएनए आइसोलेट करना है तो उसे सेंट्रीफ्यूज कीजिए, and they used a centrifugation in a cesium chloride density gradient. तो cesium chloride में डाल के डीएनए को आइसोलेट किया जा सकता है. Now they transferred the cells into a medium with normal N14H4Cl, and what did they do? Sir, N15 बैक्टीरिया के साथ N14 मिक्स किया गया. N14 was mixed with N15. And what did they observe? They observed DNA was extracted from the culture one generation after they had transformed or transferred N15 to N14, that is after 20 minutes. E. coli divides every 20 minutes, had DNAs with hybrid or intermediate density. And after 40 minutes, second generation, the there was equal amount of hybrid DNA and light DNA. So, sir, what is the meaning of this? Sir, ये है cesium chloride के अंदर N15 N15 original strand. N15 N15 original strand डाला, so they took equal with N15 N15, put it in cesium chloride and allowed it to undergo centrifugation. The DNA settles at the bottom level. DNA bottom level पे settle हो जाता है. And then they mixed N15 with N14. And after 20 minutes, they found that the DNA settles somewhere at a higher level. तो पहले bottom level पे था, अब higher level पे आ जाता है. They allowed it to replicate for another 20 minutes. After 40 minutes, they found there is at a lower level or mid level as well as a higher level. So check this here. This is lowest, this is middle, and this is middle plus highest. तो तीन लेवल के ऊपर आपको डीएनए मिला है. लोएस्ट लेवल पे मिला है, मिडिल लेवल पे मिला है, हाईएस्ट लेवल पे मिला है. इफ लोएस्ट लेवल पे मिला है, इट इज N15 N15. If it is at the highest level, it is N14 N14. That means at the middle level, it would have been N15 N14. तो लोवेस्ट लेवल पे 15-15 है, मिड लेवल पे 15-14 है, हाईएस्ट लेवल पे 14-14 है. तो ये आपको मिला सेंट्रिफिकेशन करके, 15-15 लोवेस्ट मिला, 14-14 हाईएस्ट मिला. तो बीच में कौन सा मिला रहेगा? बीच वाला डेफिनेटली मस्ट बी N15 N14, इसीलिए बीच में आया. एंड दैट मीन्स दिस इज N15 N14, this is N15 N14, N14 N15, and this is N14 N14. तो ये वाला कौन सा मिला है? 15-15 लोवेस्ट लेवल पे. ये वाला कौन सा मिला है? 15-14 मिड लेवल पे. ये कौन सा मिला है? 15-14 मिड लेवल पे. और ये कौन सा मिला है? 14-14 हाई लेवल पे. तो लोवेस्ट लेवल पे 15-15, मिडिल लेवल पे 15, लोवेस्ट लेवल पे 15-15, मिड लेवल पे 15-14, हाईएस्ट लेवल पे 14-14 एंड मिड लेवल पे 15-14. तो how would have that replicated? Sir, check this carefully. If this is 15-15, then after 20 minutes they got 15 के ऊपर आ गया 14 and 15 के ऊपर आ गया 14. Then both DNAs are hybrids. This is after 20 minutes. After 40 minutes, they get 15. They get 15 को मिला 14. 15 को मिला 14. 14 को मिला दूसरा 14. Similarly, this 14 को मिला दूसरा 14. And 15 को मिला 14. So, what is 15 getting? 15 gets 14. What does 15 get? 15 gets 14. What does 14 get? 14 gets another 14. What does 14 get? 14 gets another 14. So, after 40 minutes, you have two hybrids and two lights in a ratio of 1:1. Achha, if you multiply after 60 minutes, then what will we get? 15 will get 14. 15 will get 14. But 14 will get 14-14. 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 And 14 will get 14-14. तो दो बन गए हाइब्रिड, यहां दो हाइब्रिड थे और बीच में दो लाइट थे। यहां 60 मिनट्स के बाद दो हाइब्रिड है और बीच में 1 2 3 4 5 6 6 lights. So you have two hybrid and six light in a ratio of 2:6 or 1:3 ratio. 1:3 ratio. If we multiply for 80 minutes, then 15 will get 14. 15 will get 14. And all the other 14s will get 14 14 14 14 14 14 14 14। तो अब करो, यहां एक का बना दो, दो का बना चार, चार का बना आठ, आठ, तो आठ का बनेगा 16, आठ का बनेगा 16। एंड ऑफ दो 16, two will be hybrids. This is hybrid and this is hybrid. बीच में all the remaining 16 will become light. So two hybrids and two are hybrids, two are hybrids and बीच में 14 of them, I'm sorry, 14 of them will become light. तो total 16 मिलेंगे जिसमें से 2 you get hybrids and 14 you get light. So you get a ratio of 2:14. 2:14 ratio, which turns out to be 1:7 ratio. So 15-15 gets 15-14, 15-14. You get two hybrids after 20 minutes. The E. coli replicates every 20 minutes by semi-conservative method. After 40 minutes, you get 15-14, you get 15-14, and you get 14-14, and you get 14-14. So you get two hybrid, two light, 1:1 ratio. After 60 minutes, एक 15 को मिलेगा 14, एक 15 को मिलेगा 14, and all the others get 14-14. So you will get total 8. Total 8. 1 will become 2, 2 will become 4, 4 will become 8. Of the 8, 2 are hybrids, 2 are lights. Now 8 will become 16. 8 will become 16, of which this is hybrid, this is hybrid. And बीच में all the 14 are lights. So you get 2 hybrid and 14 lights. So ratio of 1:7. So what do we have here? Here we have heavy. Here we have two hybrids. So they are hybrids. Here we get two hybrid, two light in a ratio of 1:1. Here we get two hybrid, six light in a ratio of 2:6 or 1:3. And here we get two hybrid and 14 lights. In other words, 2:14 ratio or 1:7 ratio. तो ये कंटिन्यूसली क्या प्रूव हो रहा है? The DNA is replicating by the semi-conservative method of replication. ये नीट में बहुत बार पूछते हैं, what do we get after 20 minutes of 15 multi in 14 medium, two hybrids? What do we get in after 40 minutes? We get two hybrids, two lights. What do we get after 60 minutes? We get two hybrids, six lights. What do we get after 80 minutes? We will get two hybrids and 14 lights. तो समझो 80 minutes का 100 minutes किया, 100 minutes किया hypothetically. Then total DNAs कितने मिलेंगे? 16 थे, 16 will become 32. 16 will become 32, of which 2 will remain a hybrid, 15-14. 15-14. And बीच में 30 will become light. 30 will become light. So ratio you will get 2:30 or 1:15 ratio. 1:15 ratio. So here we have heavy. Here we have two hybrids. Here we have 1:1 ratio. then we will have 1:3 ratio, then we will have 1:7 ratio, then we will have 1:15 ratio, and so on. So whatever they ask you, after 20 minutes क्या मिलेगा, 40 minutes क्या मिलेगा, 60 minutes क्या मिलेगा, 80 minutes क्या मिलेगा, 100 minutes क्या मिलेगा, or they may ask you what you get in the first generation, second generation, third generation, fourth generation, fifth generation. Whatever they ask you, remember, you will get heavy, you will get hybrid, you will get 1:1 ratio, you will get 1:3 ratio, you will get 1:7 ratio, you will get 1:15 ratio, and so on. So this was the semi-conservative method of replication given by Messelson and Stahl. Another similar experiment using radioactive thymidine to find out the distribution of newly synthesized DNA in chromosomes was performed on Vishya Faba, Faba beans by Taylor and colleagues in 1958. तो रेडियोएक्टिव थायमिडीन से डिटेक्ट करके विश्या फैबा में टेलर एंड कलीग्स ने सेमी कंजर्वेटिव प्रूव किया that DNA replication is by the semi-conservative method. So nice experiment. Remember this one. And then there were those experiments proven as early as 1928 that something helps in passing of genetic material from one organism to the other. What that something was not known. If you remember, Mendel had found something which helps in passing from generation to generation, which he had called factors in pea plants, which we're going to do quite soon. And that was, of course, way, way before. And it was in 1928 that a British physician, Frederick Griffith, did his experiments on Diplococcus pneumoniae. डिप्लोकोकाई टू कोकाई नाम का एक बैक्टीरिया होता है डिप्लोकोकस निमोनिया दैट कॉजेज निमोनिया एंड निमोनिया से डिप्लोकोकस निमोनिया का एक कनेक्शन होता है द बैक्टीरिया कॉजेज निमोनिया ऑफ द लंग्स ड्यूरिंग हिज एक्सपेरिमेंट्स व्हाट डिड ही डू he took Diplococcus pneumoniae and grew it on culture plates and found out there are two types of bacteria. There is Diplococci like this and Diplococci like this. One type of Diplococci had a smooth polysaccharide capsule around it and capsulated tha, and there was one type which had no capsule around it. Capsule नहीं था and जो नॉन कैप्सुलेटेड वैरायटी थी उसको ग्रिफिथ ने अगार अगार सोल्यूशन या अगार जेल पे ग्रो किया और अगार जेल पे ग्रो किया द नॉन कैप्सुलेटेड वैरायटी grew, multiplied, and produced a rough colony. Rough colony, which is called as the R-type bacteria. और जो कैप्सुलेटेड स्मूथ वैरायटी थी उसको अगार पे कल्टीवेट किया it formed a smooth colony and hence it was called as the S-type bacteria. So R-type is non-capsulated and forms a rough colony. S-type is capsulated and produces a smooth colony. He took the S-type bacteria and put it into mice. Mice के अंदर S-type bacteria डाल दिया. He found the mice developed pneumonia and died. Hence, S-type is pathogenic. It causes pneumonia, pathogenic, and is called as virulent. It's a pathogen. It causes death. It's virulent. Why, sir? Because the S type had the capsule and our WBCs tried to phagocytose it, but because of the capsule, the S type cannot get phagocytosed by RWBCs, and it causes death and pneumonia in the lungs of the mice, and the mice will die. So when S type is injected into mice, the mice die. Then he took the R type bacteria and injected into mice. They did not develop pneumonia, hence R type is avirulent or non-pathogenic. R type के बाजू में capsule नहीं होती है, so when it is given in the body, RWBCs will come and phagocytose it. So R type मर जाएगा. So it will not cause pneumonia because the WBCs have phagocytosed it. So it is non-pathogenic, which is called as avirulent. So R type injected into mice, the mice remain alive. Griffith was able to kill the bacteria by heating them. So he took, say, the S type bacteria, which is smooth colony, put a burner below it, and heated and killed the bacteria. At a temperature of around 70 degrees Celsius, the enzymes inside the bacteria degenerate. If you remember, RF sir has taught you above 60 degrees Celsius, enzymes undergo denaturation. So, bacteria ke enzymes which help it to multiply undergo denaturation, and the S-type bacteria are heated and killed. So, how do you know it, sir? After a few days, size of the colony should increase, but they found size of the colony बढ़ नहीं रही है. Size of the colony is not increasing. So bacteria have stopped replication. And in the third experiment, heat-killed S-type bacteria was injected into healthy mice. They did not cause disease, and the mice remained healthy. So S-type heat killed when injected into mice, the mice remained alive. So live S-type kills. Live R-type does not kill. And killed S-type does not kill. So live S-type kills. Live R-type does not kill. And killed S-type does not kill. Now what did he do? He makes the live R-type and the killed S-type. So he took an agar-agar jali और उसके अंदर there was live R-type. उसके ऊपर he mixed the killed S-type bacteria. Killed S-type bacteria were mixed with it. So you have live R-type plus killed S-type. Killed S-type. Live R-type plus killed S-type. Now he mixed it and took live R-type, killed S-type, and put it in the mouse. Will the mice die? Sir, we have already proven live R-type does not kill. Live R-type does not kill. It is avirulent or non-pathogenic. Killed S-type, so anyway will not kill because the bacteria are not multiplying only. But to his surprise, he found when live R-type were mixed with killed S type. Live R type was mixed with killed S type. S type killed के साथ live R type mix किया. When live R type was live and it was mixed with killed S type. Killed S type. And when this was given to mouse, when this live R type and killed S type was given to mouse, the mouse developed pneumonia. S type heat killed, live R type was injected into mouse, mouse developed pneumonia and mouse died. And we have already proven that live R type does not kill the mice. Killed S type does not kill the mice, but live R type and killed S type, when mixed together, cause death of the mouse. तो ये कैसे हुआ? Live R type does not kill, heat killed S type does not kill, mice remain alive. But जब दोनों को mix किया गया, then the mouse died. And how did the mouse die? So, when blood sample from the dead mice was taken, colonies of live S type बन गया. So, from a mouse which was dead, उसके अंदर से blood निकाला तो live S type की colony मिली. तो सर ये कैसे हुआ? Live was R type, killed was S type. But when they were mixed, then something from the killed S type must have mixed with R type, and killed S type ka something must have gone to live R type, and that some particle must have transformed R type into S type by forming a capsule around it. So we have again live R type mixed with killed S type. Then from the killed S type, something must have transformed R type into S type by forming a capsule around it. What is that some particle? That some particle Griffith did not know. He called it as the transforming principle, and thus Griffith gave the first gene interaction. Griffith did not know what the transforming principle was, but we know from the killed S type, उसके अंदर था डीएनए और उसमें से जीन रिस्पॉन्सिबल फॉर कैप्सूल फॉर्मेशन. हीट करके बैक्टीरिया तो मर जाता है, but DNA does not degrade. That means the gene for capsule must have transformed R type into S type by forming a capsule around it. By forming a capsule around it, and this was called as Griffith's experiment on bacterial transformation. Griffith's experiment of bacterial transformation, and that particle was called transforming particle. This was among the first experiments to prove that there is something called as a transforming particle given by Griffith.